And as we talked about, we are honored to be a major sponsor of PBSS. So again, thank you for the opportunity for me to share who we are, what we do, and hopefully why that matters to you guys. So just as a sort of broad overview, we are a Bay Area CRO, so we are a contract research organization. We are located right here in the Bay Area, across the Bay, actually in Alameda. I would encourage each and every one of you to please come by, reach out, come visit us. We'd be happy to give you a tour. We do specialize in both small and large molecule bioanalysis. We are GMP certified. We are FDA registered, FDA compliant, and we do have a DEA license as well. There won't be any surprises for my next few slides, obviously. Really one of the biggest differentiators, I think, for ourselves is that we are very fast, both in turnaround time as well as in project initiation. We do like to think of our work together as sort of a partnership where we are very communicative, but really our mission, though, is innovative solutions for scientific problems. So really, the main questions you guys probably have are, what exactly do we do? And honestly, why you guys should care, right? Why does it actually matter? So really, what we've broken down here is there are three major components that we do in the context of what you guys care about. And that's going to be our quantitative bioanalysis, our metabolite ID profiling, and then some of the surrogate matrix strategies that we've employed. And bioanalysis, it's nothing that's surprising, especially in the light of what was talked about earlier today. But we do follow the ICH M10 guidelines. We have a bunch of different tandem mass specs. We offer both parent and metabolite identification through high-resolution mass spec in a variety of different matrices, again, some of which are obviously covered earlier this morning. In terms of the MET-ID, we do have HRMSs as well as NMR, which I think there's actually an interesting talk later this afternoon by, I think, Raju, if I remember correctly, who's going to talk a little bit about NMR and some of those capabilities in the context of bioanalysis. We are a one-stop shop for both integrative bioanalysis in the context of both quantitation as well as qualitative exploratory work for that MET-ID. We do have that regulatory mindset. We've been involved with FDA submissions, FDA meetings. So a lot of our data generation, it has that already in mind as we go from project start to finish. Again, we are very flexible and collaborative. Obviously, we are adaptive, as seen here, where I'm trying my best to keep talking while the slides keep moving forward. And again, I'd like to talk about a case study. This is a really simplistic slide, and I think for the sake of time, it's really just a general workflow where it's nothing that should be super surprising for everyone in the context of what we've been talking about, where you have your biological matrix, different sample preparation techniques, all of which we have experience doing, as well as different parameters for optimization on the analytical instrumentation end, whether that's on the chromatography side or on the specific mass spec side. In terms of the specific instrumentation, so I've kind of touched on a lot of these already, but we have a few different triple quad mass specs for quantitation, along with GCMS with both headspace and FID capabilities, so for any volatiles, if that's an analyte of interest. In addition to that, we do have high-resolution mass specs, so again, for any MET-ID with compound discoverer, an NMR, again, for any sort of structure elucidation. And then finally, we do have microplate readers for any LBAs or ELISA types of assays as well. So the case study that I did want to talk about in the few minutes that I do have is actually a publication. So I know a lot of times, CROs may not have the opportunity to publish manuscripts, but we actually have had those opportunities. And actually this past month, we were able to publish some of the work that we've done using the ICH M10 guidelines for validating a method for quantitating ketorolac in ocular matrices. The unique nature of this publication was that it required the use of a surrogate matrix. So if you are not actually familiar, both aqueous and vitreous humor is very rare. It's difficult to get a hold of, but we were able to actually use a surrogate matrix, in this case plasma, for that accurate quantitation. And with that, the methodology was just assessing for those specific parameters. With those parameters, we were able to do that assessment prior to actually getting real-life samples from this clinical PK study. So really, this is just a one-slide summary sort of highlighting what those actual results were. So again, all of our accuracies in both of those matrices using that surrogate matrix did fall within the ICH acceptance criteria of that 85 to 115%. Obviously, that 20% threshold for our LLOQ, our lowered limit of detection, or quantitation, excuse me. We did assess for specificity where we didn't see any endogenous matrix components, particularly for the use of that surrogate matrix and why that's important. And then this is just figures and tables taken directly from the manuscript, which really just highlight what I've summarized verbally. Here's really just an overall slide of just talking about the team, our specific bioanalysis team. So we have over a dozen different scientists, majority of which do have advanced degrees. We combined have easily over 40 years of experience in the industry, as well as in addition to the publication I talked about, we do have 10 or more where we're able to do a lot of these publications, either in journals or give presentations like the one that I'm currently giving. That typically goes slightly smoother without the auto animation here. In terms of some of the highlights, I just want to say that the biggest thing that we offer is that a lot of our study initiation rates do exceed the normal. We take pride in being able to start projects earlier. We do offer same-day sample analysis under specific circumstances, but in general, we turn around things within 2 weeks. And again, all of those sort of instruments that I talked about, we do have backups for. And I'd love, again, to reiterate that if any of you want to come visit our facility, we'd be more than happy to have you. So if there's anything to take away from this presentation, besides the fact that obviously there was animation here that I wasn't aware of, is that we offer both integrative, quantitative, and qualitative workflows. You know, we do offer, again, that sort of fast turnaround time. We think of our relationship as a partnership, so we will work with you. We'd be happy to go through the data with you, team you, and do whatever it takes to make sure that you guys are happy. We do have that experience in those rare matrices that I talked about. And again, this is just sort of a workflow summarizing the different types of instruments we offer and some of those capabilities. You know, I thank you for your patience, and I know hopefully none of you got a headache or seizure from me going back and forth. But you do, wow, it's really flying. But you do have the PDFs of those slides. If anything was unclear, please come out. We are right outside here to the left. I would be happy, or anyone on our team would be happy to chat more about any of these things. I appreciate your time and understanding throughout this presentation, so thank you.